Hi, hello everyone, Microbehunter here, and welcome to another Saturday live stream. And today I would like to do a little bit of a stereomicroscopy. I do have a new stereo microscope, and it's a very nice one that I have, and I would like to use it today to show you a few of those specimens that I'll have over here. And I hope that it's going to be interesting for all of us. Well, I see that many people already started watching, and I would like to, as always, not only send a heartfelt welcome around the world, but of course I would also like to, kind of like to know if you're able to hear me properly, if the sound is fine. Maybe you can write a quick comment into the chat section here. Okay, so this is something that. I see that there are now quite a few people again. Yes, I hear you. Thank you very much. You see, every time when I start the program again, I have to readjust the audio settings a little bit, and sometimes the audio is a little bit too loud. Sometimes people have problems hearing me, so that's why I have to ask you. It is going to be like this, that for those of you who are new, it's going to be like this, that the live stream will last somewhat over an hour, sometimes 1.5 hours. Sometimes I will interrupt myself a little bit to respond to some of the questions. The rules are as follows. You can, of course, post any kind of questions that you want, as long as they're microscopy related, and even if they're a little bit off topic, as long as they're microscopy related, I would like to answer them. However, there has been also a request in one of my previous videos that I do not interrupt the live stream too often to answer questions. So what I might do is I might simply show you a few things, and then after a couple of minutes I might interrupt myself, and then I'm going to read again some of the comments. But at the beginning, what I would like to do is because this has become a little bit of a tradition, I would like to quickly read out from where you are from. So there's a hello from Vietnam, there is a hello from the UK, somebody is waiting for the Journey to the Microcosmos microscope. That's another YouTube channel, and they're also selling microscopes. And there is a hello from all over the world, somebody's afraid of spiders here as well. I will talk a little bit about those specimens here. So hello from Massachusetts. We're from the United States. Okay, and there's a hello from Finland, and so on. So what I will do is I will simply hello from Norway, Bulgaria. Okay, so that's very nice. So a short introduction to what we'll be doing today. First of all, maybe one of the things that you have noticed already over here is this gigantic spider here. Let me assure you, this is only the exoskeleton of the spider. So in other words, only the skin. You don't call it skin, but it's only the exoskeleton, because the spider molted. Where do I have this from? Well, I'm a biology teacher, and in our biology storage room we have a lot of different specimens that we collected over the years, that we have some demonstration objects for our students, and this is where I borrowed it, so I have to return it again when I've used it. And it's again, it's only the skin, and there is a glass lid on top. It's taped closed, and I will be able to put it directly under the stereo microscope as is, because it's kind of delicate and fragile, so I don't want to open it. And then you can also see a whole range of commercial insects that are basically in this block of plastic or resin, I would rather say. There's always a little barcode here also on the side, and these are commercially, but the real ones, not only insects but also scorpions. They're related to the spiders as well, and they have an advantage as well, that you can also put them directly under a stereo microscope. And one of the reasons why we got those is because, again, these are fascinating demonstration objects that children in school can use to practice a little bit of microscopy, because we do have not only compound microscopes but also stereo microscopes. And this is the reason why we got those. So there are quite a few of them. By the way, if you are interested in stereo microscopy, I do have another channel as well, called Microscopic Mysteries. It's not a very active channel, but there I put objects under the microscope, and then you can guess what they are. So it is not a narrated channel, but it's simply one that I started some years ago. But it's not very active. But you can try to visit it as well. Microscopic Mysteries, I call it. And because I wanted to have a channel that's dedicated to stereomicroscopy as well. And look at this gigantic fly over here, which that's of course not cast in resin, but I'm also going to put this under the microscope here as well. And some of you might be a little bit disappointed that all of those insects and spiders are not alive. I do have 3 over there on the side. I've got 3 ant lions that are caught today. Just before I started the live stream, I caught some ant lions. They're running around in my little petri dish. I would like to show them to you as well. Okay, I would like to now switch over to the stereo microscope view. So let's put the ant lions away. Let's save them a little bit for later so that you have a little slight motivation to continue watching. And I would like to switch over now to the stereoscope. And every time when I do that, I have to quickly switch on the camera as well. Okay, here we go. And what you're able to see now here is in the corner, somewhere over there, all the way in the far corner. Actually, I do have an arrow. Okay, here now we have got that's the wrong one. Now I'm moving the camera around. I don't want that, but I want to move the arrow around. Yes, here. That is basically that's the stage of the stereo microscope. And then in the background, you're actually able to see the magnified in image. So whatever I put on here, like for example this insect over here, then we're able to watch this. I would like to talk a little bit more about the stereo microscope. It's a new one that I've got now. It's called the Nexius Zoom from the company Euromex. Those of you who have been watching my other YouTube channel know that I've in the recent weeks been doing quite a lot of review videos on Euromex microscopes. This one is a new one that I've got now. It's one with a continuous zoom. So you can actually see that you can zoom in. Okay, I have to focus as well. And it's actually the first time that I'm using this microscope for a live stream. Okay, it's a camera connected and I have a continuous zoom here. And just a side information, what I'll be doing is I'll be making a separate review video of this microscope as well. So I don't want to talk too much about the microscope right now because there will be a separate video on it. I'm just going to show it to you. So basically what I planned to do today is no advanced science, no advanced specimen preparation or anything. I just want to show you a couple of these specimens to have a little bit of fun. Okay, but before I actually get started, already in last week I already made a little announcement here, and I have to switch back now. No, or just hold it up like this. Okay, I would like to motivate hobby microscopists to start making videos. This is the picture of Anthony van Leeuwenhoek. He was actually one of the, if not the first microscopist around, and 2023 is the 300-year anniversary. He was actually the guy who first saw bacteria and microorganisms. He made microscopes himself 300 years ago, and he was very successful with that. And I just recently discovered that we have the 300-year anniversary, and you just said, well, actually, why not use this opportunity to make videos? So you don't send the videos to me or anything like that. You've got a YouTube channel. If you've got a Gmail address or Google address, then you already have a YouTube channel. Just make some videos, share your knowledge, your experience, and put this hashtag into the title or somewhere in the description. And with this hashtag, if anyone who Googles for this hashtag will then find your videos. I think this way I would like to motivate other people who are into microscopy to start making videos, and maybe we can share some of the knowledge. Even I myself learn a lot by also watching videos of other people. So that's basically. I see the chat is already going. Wow, overflowing here. Okay. And there is still a lot of hello going on. So what I will do is I will simply show you a few of these specimens here. I think here on the side, next to the barcode, I have to, maybe we're able to, I have to refocus this again. Maybe we're able to read what it actually, See the image is flipped over, unfortunately. Okay, that's pretty sad. Okay, I didn't realize this. Okay, that the image is actually flipped, so I have to made in China. You can see. Okay, yeah. So but there's also a web address here, so maybe what I have to do is I have to flip it horizontally, but I think it'll just leave it right now as it is, and I have to refocus this, and there is, in other words, these are pretty gigantic insects. I think they must have been bred and specifically made for that purpose. What we're gonna do is you see that the video is a little bit slow, but that is always the case with USB. And one thing that stereo microscopes are able to do is they're able to produce a stereoscopic three-dimensional image, and that's of course something that unfortunately we're not able to see here. I would like to, in any case, promote stereomicroscopes a little bit, especially if you are considering microscopy with children, with young children especially. Then you do not want to do a lot of complicated specimen preparation, but rather you're probably interested in simply observing nature in a very direct and uncomplicated way. You just want to take some leaves, some insects, some whatever, and put it directly under the microscope. And with stereomicroscopes, you're able to do that. Okay, and this stereomicroscope is able to magnify, I have to check now, from 6.5 times to 55 times. Okay, so I have a zoom functionality here as well. It's a continuous zoom. So what I'll do right now is I'll just start to zoom in. I might have to refocus every now and then, but I just wanted to show you a little bit how this actually works. And I adjusted the camera in such a way that essentially it will adjust the exposure time accordingly. Yeah, because normally when you start to zoom in, the image becomes of course much darker. But here the camera will adjust the exposure time at least as much as it can do. So that's not a maximum magnification that we get. Okay, and of course at this magnification, only if you touch it a little bit, then you're able to already see that it moves quite a bit. Okay, just yesterday I had the possibility to have a short talk with an entomologist. Entomologist is a researcher who is specialized in insects, and the gentleman that I talked with, he is a specialist on ants. Okay, and I asked him, of course, which type of microscope do you have, and he said, yeah, he has also got a stereomicroscope from the company Wild, which has been bought up by Leica later on. And he said he needs magnifications over 100 times with a stereomicroscope, so he needs to add additional lenses to it. And I said, why do you need such a high magnification? And he says, well, in order to distinguish some ant specimens, you actually have to count and measure out the different hair that are growing on the ant. Yeah, so it's actually because apparently on the number of hairs that you're able to count, this already determines which type of species of ant it is. And he says you can only see that with very high magnifications. When I say very high magnifications, I mean very high magnifications for stereomicroscopes. So this one over here, the maximum magnification I have to check here is 55 times, at least using the eyepieces that I have here. Okay, so what I will do now is I'll just put in, this here, for example, is a scorpion. Okay, see, centering it is always a little bit of a challenge, and you can also see that there is a little bit of dirt, of course, on the plastic or on the resin block. Yeah. So what I have to do now again is I have to refocus this a little bit. Yeah. And now it's getting blurry, so I have to always know into which direction to turn. So what I'll do now is I'll go up and I will go down some of the questions again a little bit, and then I'm gonna show you some more here. Is there any difference between microorganisms living on people from different races? Let's put it this way. As a biologist, I have to say, how do you define a human race? Genetics actually has shown that races in the humans actually as such do not exist. It's a continuum, but actually it's most likely also possible that there are different microorganisms also living, not only on people of different races, as you say, but actually people who live in different regions of the world, because different people eat different food, and therefore the biome of a person can also be different. So you don't even have to look at the ethnic origin of people, but simply of where people live. Why not? Okay, so this is almost quite certain the case. But again, the whole race concept in biology is an outdated concept, because there is no genetic evidence, at least in humans, for the existence of so-called what used to be called races. It's a continuum that we have. Okay, so hello from Finland, Bulgaria. Is there any difference between microorganisms and people? Oh, it's the same question here again. Okay, yeah, hello from Norway. What is the most intelligent person in microscopy you have met? I cannot answer this question. I think it's not necessarily a question of intelligence, but maybe people who are knowledgeable. And I like to talk to all people from who I can learn something from. It's not so much in my case, not what I am interested in is in having interesting conversations with other people. Yeah, basically, if people know something what I don't know, lots of people know things that I don't know, obviously. I mean, and I like to talk to those people. Yeah, and therefore, it is not really, yeah, it depends really more on the topic than on the person here. Where did you get that spider? Well, yes, as I already mentioned, this gigantic spider that I've yeah, already showed you over here, this one here, is part of the collection that we have in our biology department in school. Yeah. So yeah, somebody loves my videos here. Okay, yeah, it's yeah. Hi, I did entomology in Master of Science. Oh, that's nice. Okay. Hello to everyone around the world. Have you ever killed microorganisms by pressing the objective against the slide? By pressing the cover glass against and sandwiching the microorganism? Yes, you don't even have to press it. When the water evaporates beneath a microscope slide, then what happens is, because of the surface tension, the microscope slide cover glass presses and sandwiches the organism. And this is actually, if you don't overdo that, then that is actually also a recommended method to kind of limit and inhibit the movement of certain microorganisms. Otherwise, some of them are moving too quickly. And by sandwiching them between the cover glass and the microscope slide, you're limiting the movement a little bit. If you overdo that, you actually can crush them, yeah, so that they pop open and the content of the cell runs out. So that is possible. Okay. Okay, we're all gonna become entomologists. I mean, there are a lot of people out there who actually study insects as a hobby. Okay. Have you ever used a 40 times oil objective or 60 times oil objective? No, I have not used a 40 times or 60 times oil objective yet. I know that 60 times oil objectives do exist, but I've not used them yet. And generally speaking, I do not really like oil objectives so much. Sometimes you need them depending on what you want to observe, but I think they're a little bit messy, right? And I'm kind of always worried of kind of contaminating and making the non-oil objectives oily. And then you have to clean it, and so it's for me it's a little too risky. So I don't like it so much, but I understand that there are some cases that where you should use them, right? Yeah, even though I'm afraid of spiders and other insects. Well, I have to now correct you here. Because you said other insects. Strictly speaking, spiders are not insects. Spiders and crabs and insects belong to the arthropods, so that's the category. But spiders are arachnids, and you can know the difference because among other things, the number of legs. Spiders have 4 leg pairs and insects have 6. I was looking at something in my basement and I found the same insect collection here. Yes. Yep. Yeah, good evening everyone. You changed your okay, it's for somebody else. What is the largest thing you have ever put under a compound microscope? Yes, it is possible to also observe, let's say, flies, like I have here, over here, under a compound microscope. So I have put whole insects also under the compound microscope, and it is possible to look at them with the low power 4 times objective. So what you do is you need a strong flashlight, and then it is possible. Okay, as a matter of fact, I recommend that everyone tries this. Get yourself a strong flashlight and put some objects. Start off with easy objects like, I don't know, pieces of paper with some writing on it, because focusing is easier. put it on your microscope and put the light from the top, and you're going to get similar images as you have over here. But I'm not talking so much, and let's try to simply look at a few other things here as well. Yeah. You can see that there are quite a lot of scratches already on the surface, and that those insects that we have here are already relatively large, and I'm working at the lowest magnification. However, this microscope, I'll just show you what I'm able to do. Has this thing here, so I'm able to turn it around, and now I have a white white surface, and I can also show you something else here. You can see that in here there is now I've got light coming from the top, but there is over here also an LED. And what you can do is you can put this glass over here, and that's now not the white one that I had from before, not this one, but this one I just showed you here, right? So I've got a transparent one, and I've got one that looks like this. And if you put the transparent one over it, and then you can also turn it off. You can also have light coming from the bottom, and this is of course actually I just realized this looks actually much better. Okay, cool. The light is so bright that actually the insects become transparent. Okay, so so this is basically I've not shown you so many things, but let's have a look. It's too bright, I think. So let's do this again here. So when preparing, is it possible to prepare insects also for and put them on a microscope slide? This is the kind of the question, right? And this is possible, of course, but as you can see, see so many scratches. The problem is that most insects are way too dark and too thick. So what you have to do is if you want to put an insect, a whole insect on a microscope slide, then you have to essentially compress it flat, and you have to make sure that it becomes bleached. And one way of doing that is by putting the insect for a couple of days into a strong potassium hydroxide solution, and this will brighten the exoskeleton. And then you remove the water by putting it into alcohol. Okay, and then you're able to compress it flat between a slide and a cover glass. Here of course we're looking at the external part of the insects here. So this fly over here, let's have a look at the fly. We caught a few days ago. And what I'm going to do now is I have to focus this again, and this is of course looks not much nicer because it's not in resin and in plastic. And in the thumbnail I included the same picture of this fly, and what I've done, and this is also something where I'm going to make a video, or actually I already have made videos about this, is the so-called the whole issue of focus stacking. Okay. For example, when you focus the fly, then you're now going to see that certain parts of the fly are out of focus. So what you do is you take pictures, and you always turn the focus a little bit like I'm doing right now, and different parts of the fly start to become blurry, and other parts of the fly start to go into focus. And if you take a picture of each one of them, then you're able to use a software to combine it together into one completely clear image. Okay, and this is actually done with the click of a mouse button. If you want to try this yourself, then I can recommend the program Helicon Focus. Helicon Focus, it's a commercial product, but there is a 30-day trial possibility. And then you're able to combine different pictures of a different focus into one image that is completely sharp. However, I want you to now have a very close look at the horizontal position of the fly. So what I'm doing now is I'm just turning the focus, and I'm shifting the microscope head up and down, and look what happens to the fly as I focus it. Do you see that it starts to move to the left? Okay, so I'm focusing back, down. It not only comes into focus, but now the fly also starts to move to the right. So there is this, when you use a stereo microscope, and when you start focusing, there is this horizontal shift. The question is, why is that? And the reason is a stereo microscope has two objectives, but one objective is used to actually direct the light to the camera. And this basically means it's off axis, right? And as it moves down, and if you only use the light of one objective for the camera as it moves down, then there is this apparent horizontal shift, because the angle at which it looks at the fly also changes. So this is simply something that you have to be aware of, but the stacking software will actually remove that. So for example, if I put my fingers down under the microscope in one, now I'm covering this one objective, but if I'm moving it beneath the other objective, nothing happens, because only the light of one objective is redirected to the camera. Yeah? So this is also something that that you can do, is to do a little bit of focus stacking. And because I've got this microscope now, of course I will make a couple of tutorial videos on how you can focus stack, because it's pretty simple really, because of the software that's available. And yeah, but the effect is quite nice. I don't know, maybe we can try it like this as well. I don't know if I'm able to do that, to hold it like this, and then maybe we're able to look at the fly also from the top. Yeah. By the way, I'm always using now the lowest magnification, right? With the reason that the specimens are so extremely large that yeah, I'm not able to do it differently. So let me see if I have this. Yeah, I also wanted to show you something else right now, because later on I want to show the ant lions that I promised you. I also want to show you something else, also to put a little bit of variation into the whole live stream. So let me switch over again to desk view. Okay, so let's make a little bit of something clear here. Look, I bought those as well. These are so-called Barlow lenses. So over here it says 2 times. And this one over here, I think I bought them over AliExpress, 0.5 times. And what happens now is I can connect them beneath the microscope objectives, and this one allows me to double the magnification, and this allows me to reduce the magnification. Okay, so this means I can actually go with this here to a magnification of over 100 times with a stereo microscope. Now the amount of light is already very low at that magnification, so you have to maybe add an additional light, but I just wanted to show you that it is possible to yet further increase or decrease. Now you're going to say, why would anyone want to decrease the magnification? The reason is it's probably not so much the importance of decreasing it, but with this Barlow lens, you can really move the microscope objective far away, so you have a very large distance between the object and the objective in case you need that. So this is actually not so much important only for reducing the magnification in case the object is too large, but also by increasing the distance between the objective and the specimen. Yeah. So yeah, these things can be easily connected as well. I think I'm not going to do this now, because then I have to really move up the camera quite a bit, but all of these options also exist. Yeah, by the way, in case you want to know, let me go back to the stereoscope. I'm just going to, yeah, okay, just a second. There is now no stereo. I always have to deactivate and activate the camera again, otherwise there is no, yeah, here we go again. Just now look maybe into the corner. Okay, look into the corner. Now I want to show you a little bit how the microscope actually looks like, how the setup looks like. Okay, that's the microscope here. Okay, yeah. So I've got one over here. This is a, yeah, it is so in some microscopes you have a beam here, or some kind of a bar, a pillar they call it, but I wanted to have the one where there is no pillar, but one where there's a continuous a rack, so that you can continually zoom and you do not have to loosen a screw in the back. So this is actually quite nice. I have also over here a lamp, yeah, here on the bottom, yeah, as well, pretty large LED lamp. And what you can do is, yeah, you can connect those Barlow lenses by screwing them, and that's a little bit difficult now, but yeah, by screwing it, yeah, down here. We might actually try it like this. Why not? Because the insects are so large. Let's try it like this, but it might be necessary really to, yeah, you see, yes, the magnification is lower, but you know, I have to move it much higher, I think. And I wonder if this is possible. You see, yeah, I have to, I'm a little bit out of focus now. I'd have to adjust the camera again. Yeah, so I'm just going to remove it again. So yeah, just wanted to show this to you a little bit, and now you see that the magnification is much stronger, much higher again. But again, this insect here is way too dark. Okay, so let me use another brighter one here. It's some kind of a wasp or a hornet. So what I'll do now is the following. What time is it? Oh, okay, I've been now live for 30 minutes. So what I'll do now is I'll read again a couple of comments, and then maybe I'm going to show you a couple of the ant lions which are running around over here on my table. Okay, let me scroll down here. Hello everyone, sorry I'm a bit later. Photo changed his name. Okay, there's some talk in the community going on. Is there anything you wish you could see under the microscope but can't? Is there anything I wish I could see under the microscope but can't? Well, a lot of things can be seen under the microscope. What I would like to, I mean, let's put it this way, it depends really on which type of microscope you're looking at. Nowadays, you can, it would be, I mean, this is, would be of course nice. It would be of course nice to see certain cell organelles, yet better under the microscope. Those that are difficult to see, I mean, for example, the endoplasmic reticulum and so on. But the reason is that the resolution is not high enough of light microscopes. Those membranes are very, very thin. So that would be, of course, a nice thing. Certain processes of life yet further, to be able to see them yet better. So yeah, it's so hot in here in Vietnam. Trust me, currently we are also experiencing a little heat wave here in Central Europe, where I am. Okay, so at microphone, is there a way to change magnification on an eyepiece camera? It's much more than 25 times an eyepiece camera. Well, let's put it this way. The camera that I've got up here, you know what, I'm just going to raise this again. I'm going to show you here. Okay, so the camera that I have up here has an adapter here with some optics, and those optics here are actually important for adjusting the image size so that it matches the sensor size. So if you want to adjust the magnification of anything, you'd have to change the optics here. Or a second possibility is that you use a camera with a larger sensor. This will also reduce the magnification. So you see, it's a combination of multiple factors. But because this is sometimes not a practical solution, I recommend that if, for example, you want the object is too large and you want to get the whole object on the camera, that you do so-called image stitching. So what you do is you take overlapping pictures of your image, and then you combine them into one larger image using panorama software. One software that I recommend, it's not, you have to search for it, because it's not maintained anymore, is a Microsoft ICE, Image Composite Editor, MS ICE it's called, and this is a very good panorama software which allows you to combine images in 2 dimensions. So this is so this is kind of a workaround if you're not able to change the magnification of the eyepiece camera, that you're able to do this indirectly by stitching together pictures. Okay, is it okay to do an agar petri dish of the microbes that live on my feet? And the sure answer is no. Okay, don't do that. Microorganisms that are living on your skin are potentially pathogenic, potentially disease causing. They are adapted to live on human skin. And if you now start making agar plates where you're growing bacteria that have the possibility to grow on human skin, that's not a good idea, because if you have a wound and if you accidentally get those, you touch the plate and you're getting those bacteria into your body. I mean, this is a crazily high concentration of bacteria. Yeah, so don't do that, okay? So that's why I'm not really happy on YouTube when I see that people actually make those fingerprints on agar plates, and so I'm really not happy. Even if you make a fingerprint, maybe there is already a contaminant on the plate, right? And you're transferring the billions and billions of bacteria on your skin, and you don't know what they are. Huh? So this is not something I recommend. If you want to work with bacteria, I highly recommend that you use yogurt bacteria. And that you make yogurt at home. Okay, yeah. So, and that's also one of the reasons last week when I made my live stream with the E. coli bacteria. Yeah, I've done this because yeah, for educational reasons, and I was not growing unknown bacteria here. Yeah. So, is there a way to change the magnification? I'd already talked about that. I think I caught the same fly a few days ago. Okay, Rinderbremse. I don't know. I have not tried yet. I have not yet tried to identify it, okay? Why so much bass in the audio? I don't know. Should I tell me a little bit, maybe because it's next to my mouth. Maybe if I move it a little bit further away, the microphone, maybe there's going to be a little bit of less bass here, okay? What's the most expensive monocular microscope you have ever seen or heard of? Well, I don't know about the monocular microscope. Yes, oh yes. This is actually a topic that I wanted to make a separate video on. So Wambi has asked a very important question. What is the most expensive monocular microscope? That's a microscope with one eyepiece that you've ever seen or heard of. And I've seen them on Amazon, and I was really upset because these were introductory microscopes which cost around, when you buy them normally around, I don't know, 60 to 100 euros or dollars. Not much more than 80 or 90. And it was sold for way over $1,000. And on several, the product was listed several times. And this was I think what I'll call this a scam, okay? I think it was not an accident because the microscope was listed several times. With an outrageously high price, and yeah, I think this is not okay. Yeah, I think the equipment should cost what they should cost, but not unnecessarily more. Okay. So, what kind of insect is it? I don't know if this is the one that it looks like a hornet, yeah, or some kind of a wasp. Unfortunately, I'm not able to read it. What it says here. Okay. So. What I would like to do now is the following. I would like to show you some of the ant lions, and I will do that by first showing you a little video that I made just a few hours ago, and that's the video over here. Okay, so this is outside in the front door, and there are between the rocks there are those little funnel shaped in the sand, those little funnel shaped structures, and these are, and there's an ant fell into it, and you can see that the ant lion, which is hidden in the sand, starts to throw sand at the ant so that it keeps on falling back into the funnel, and then the ant is being grabbed and is pulled into the sand by the ant lion, and then it's eaten up. Well, actually, the ant lion does not eat the ant completely, but it actually sucks it out, the contents, and then throws out the empty body. And so what I've done is I've taken a spoon and I tried to get the ant lion out. A little bit difficult because it had pretty much the same color as the sand. Okay, and so I was digging around a little bit and I was able to scoop out the ant lion here. And the ant lions are larvae of certain insects. These are the larval stage of an insect. And so what I've done, I've actually caught 3 of them and I put them into the Petri dish. But now you would probably like to see how those, how does the insect look like? Yeah, here again, it's looping it, so kind of fun, not so fun for the ant. But the ant lion is throwing sand at the ant so that the ant always falls back in and it's sucked in. Yeah? Like in a nightmare. Okay, I just want to show you now this here. It's a public domain picture of the adult insect of the ant lion. It looks totally different. It's fascinating. Yeah, but that's what insects do. They undergo metamorphosis. So yeah, they change from a larval stage to the adult stage, and it looks totally different. Yeah. So I mean, I already consider this quite fascinating. That So imagine that all of those insects that I have here, okay, that they will actually have a larval stage which looks completely different. So this is kind of the thing here, which was kind of interesting. And I'm going to now show you. I'm going to switch over now to my desk view again. Here it is. So let me clear this off. And here on the Petri dish, this is where I have got the 3 antlions here. Yeah, you see it's running around a little bit. So there's a little bit of sand left in here. Yeah, of course now they felt that I was touching it, and here it's moving again. And later on I'll put it back into the sand. I've got a little jar over there with some sand, and then I'll put it back where I got them from. You see that they're kind of running around a little bit, and I think we're just going to put it under the microscope, as always. So I go against stereoscope. I have to reset the camera all the time, and let's have a look now. Okay. Of course, moving objects are a little bit more difficult to. Here we go. I have to kind of refocus this. Here we go. See, it's still covered in sand a little bit. You can see this gigantic, I don't know, pincers or claws or whatever you want to call them. Here's the second one on the side as well. You have to kind of always move it back into the. Almost looks like it's kind of moving backwards always. It's the smaller one of those. I don't know, maybe it responds to the light a little bit. Maybe it doesn't like the light and tries to move away from the light. I don't know. But here's the 2 bigger ones over here. Almost like aliens they look. Again, these are the larval stage. The larval stage of this insect. So I don't know, maybe I'm just going to try to zoom in a little bit more. See, that's makes it always a little difficult, of course. You can also see the eyes even. Let's focus again a little bit. That's actually, if you want to observe insects under a stereomicroscope, strictly speaking, you have to kill them. Otherwise, in most cases, they fly away or they might sting you. And as you can see, especially because of the magnification, it's kind of difficult to center them. Maybe, I don't know, maybe like this. Maybe if I just keep them like this here. Let's put it here. But it could be that maybe it runs upwards. Finding it is difficult. The magnification was too high. Here, okay. I tilted now the slide, so now they have a little problem of moving away. And as we watch the antlions, let's have a look at a few more questions here. Is there a way to change the magnification of an eyepiece camera? I mean, what you can do is you can zoom in, of course, and as I said, is you can do some image stitching. I like the color of the insect. Cool topic for today. I have a rhino beetle, which is put in glass. I never thought of putting it under the stereomicroscope. Yep. Well, do you also have a stereomicroscope? I don't have one. Okay, that's basically, again, communication inside the forum. Would it be possible to produce 3D images from stereomicroscopes, like the ones you see with color 3D glasses? I guess it would need to do identical cameras. What you can do, I've never tried this, okay? It should be possible. What you could do is you could simply connect basically a mobile phone and take a picture through the left eyepiece and through the right eyepiece. That would be one possibility. Alignment is, of course, always a challenge in this case. Or maybe a second possibility could be that you simply shift the object horizontally a little bit. Okay, So that means if you take a picture using the trinocular port of the microscope, you simply shift the object a little bit left, and you shift it, take a picture, you shift it a little bit right, and then you might be able to do that. If you present both images on the screen, and if you practice a little bit with crossed-eyed viewing, I tried this, then indeed this is possible. There is also a second way how you can make stereoscopic images, and there is a free program called Picolet. Picolet.de. I might actually type it in here. Picolet.de. Enter. Okay. So this is basically the web page, and there you can do image stacking, just like I told you before. But this program Picolet is also able to, from the data, to actually generate images that can be viewed using those red-green glasses. Okay. However, the program Picolet is a little bit advanced to be used. Okay, but it is possible. So yeah, so that's the 3D glasses one. Okay, yeah. So again, some general communication. I got it 3 in total. Okay, that's a lot. I've got 1 compound microscope. Scorpions, yes. Hello and good evening from northern Germany. Yeah, I want to see the scorpion's tail. Okay, well, let's have a look, because we know that the scorpion's tail is a little bit poisonous. Okay, so I've got 2 scorpions here. Okay, but I think the tail over here is better to see. So yeah, let's put the antlions away again a little bit, and let's have a look at this part of the scorpion's tail, which yeah, is the dangerous part, so to say. I have to see again. See, I have to. There is a little bit of a delay in. Here we go. Here. Look at this. So that is the tail of the scorpion. Okay, and that is the pointed part. Wow, look at this. Let's go in here a little bit more. Yeah, look how sharp and pointed it is. Let's focus. Maybe I need a little more light. Okay. Or maybe I'm going to turn the light on from the bottom. Let's try this. Maybe the bottom. So now I've got bottom light. Yeah. Let's go a little bit higher yet with the map. Oh, it's the wrong direction. So, okay, I'm a little bit also off center. Okay. So that is, yeah, I wonder if this is some kind of a tube or canal in there for the poison. That would be kind of interesting to see. It's too dark. I don't know, maybe it was a little bit. So that's the maximum magnification that I'm able to get now. Focus. It's pretty sharp. Okay, I don't know. I'm not able to see any. Yeah. So, but that is basically the. Yeah, let's go out a little bit with the magnification again. That is the tail of the scorpion. See, the camera always will adjust for the exposure, but sometimes maybe it's better to have light coming from the top. Okay, that's light from the top. Okay. Have you ever seen the lollipops with real scorpions inside? Oh, I heard about those. Okay, but I don't know how to get them. Okay. Yeah, so somebody has got 2 amscopes. Okay. Groups of people who live in the same climate and have the same food source. Okay. Omax broke, amscope arrived broke. Ooh. That's sad. Okay, if microscopes then are damaged when they're on arrival, and because they've not been properly packaged, you gotta send them back. Yeah. Is there a way to change magnification on an eyepiece camera? It's much more than 25 times. So you want to make it more than 25 times? So I mean, again, what you can do is there is no easy way. What you can try to do to change the magnification is to move it in and out. And further away, the further away you move it, the larger the image becomes. But then it's not parfocal anymore. Yeah? So this is indeed something that I cannot really say that there is no easy way to do that. If you want to reduce the magnification of a camera, yeah, then you have to basically take overlapping images and join them together, and then you have essentially a lower magnification in the image. Yeah. I have a Swift stereoscope with a 10 times and a 25 times eyepiece. The 10 times is amazing, but the 25 times sucks. Field of view is so tiny it only fills a small circle. Yeah, usually with lower cost microscopes generally, the 25 times eyepieces usually aren't that good, because also the eye relief is very short. This means you have to go move very close to the front lens to actually see something. How do you get a such clear image considering the refractive index with glass? I suppose that you're asking how is it possible to get a relatively clear image even though there is a resin on top of the specimen. The reason is that first of all, the effect of something in between is stronger the higher you magnify something. Yeah? But what we have here is that any dirt or any scratches on the surface of the resin are so much out of focus that you do not see them. Yeah. So I'm basically focusing through the first couple of millimeters of the glass. Yeah? And that's the reason why I'm able to get a reasonably clear picture. Okay. So let me go down again. It's a type of Heteroptera. Yeah, so basically, yeah, I think it's these two here. Heteroptera are those, how do you call this? Where's, I mean, these ones over here are not so well. Depending on whether the wings basically are completely sclerotinized or not, basically hardened or not. Yeah, here you can see the scratches on the surface. Yeah, you can, yeah, that's basically a bug. Okay, true bugs, never heard of that before. Yeah, okay, let me talk about, how do you say, Hymenoptercoleoptera. Okay. Let me talk about this here as well. So let's do a little bit of science, because why not? Because some people wanted to know that. So let's talk a little bit about the different types of insects that we have here. Okay, so I'm going to move all the way down here. And what do we have over here is the following. When you look at these things here, then you have those beetles. They have two pairs of wings. Okay, and the top wing, the front wing is very hard. Okay, and the front wing is very hard and colored. And then when the insect spreads the front wings, then the hind wings, the second pair of wings, are very thin. Okay, so these are basically the beetles have this type of thing. For example, if you think of a ladybug, then the red wing with the black dots, yeah, that's the front pair of wings, and it kind of flips it away, and then beneath it you have the flying wings, okay, the second pair. Yeah, so that's one type of insect. They're the Coleopteras. Then you have the Heteropteras, and the Heteropteras also have two pairs of wings, but the front wing is hard the half of the way and membranous on the lower half. That's why it's called hetero, means other. Yeah, so it's not continuously solid, the front pair of wings, but only half solid. Yeah, and then you have the Hymenoptera, which has two pairs of wings, but both of them are thin and membranous. Yeah, so it's kind of a little bit difficult to show this to you because I'm not able to spread the wings of these insects here. But essentially these are different categories depending on the wings. Yeah, so you normally don't think that beetles have wings, but actually they have two pairs of wings as well. Yeah, the Coleoptera. So these are the bugs. So these are the true bugs. So I say are the Heteropteras, and then you have the Hymenopteras, which are the bees, for example. Yeah, and so on. Yeah. And then you have the flies, for example, are known as the Dipteras because they only have one pair of true wings. I think this is actually an interesting topic, but I would say almost would fill a completely separate video here. Okay, yeah. So yeah, So you have so many insects that I cannot tell which one is which. I'm not an insect expert either. Well, honestly, I'll be very honest with you. Neither am I. There's so many different thousands of insects out there. And one has to probably read the instructions or the descriptions over here. Okay. My favorite semi-living thing is the bacteriophage, which is a virus which infects bacteria. Okay. What's your rarest slide? Well, actually, I don't have many rare slides, honestly. Each slide is unique a little bit, but I do not have any expensive or rare slides, I have to honestly tell you. I mean, I have a whole bunch of commercially made slides, but ultimately those slides that I have is, none of them are very rare. Some people are into slide collecting, especially slides from the 19th century or collector's items. And there can be some rare ones there as well. Yeah. If you show the green metallic beetle again, can you zoom in and show the details from its carapace? I think it's on those beetles whose surface creates the color. Yes, this is indeed the case. I think it's this metallic beetle. It looks green, but in reality, it is not it's not colored green, but it is because of the light interference why it shines metallic green. Okay. Let's zoom in a little bit. I've not tried this myself yet. There are a whole bunch of scratches here also on the surface of the resin. I have to refocus. You see, that is actually one of the mistakes that I've done is I should adjust the microscope in such a way that I do not have to refocus when changing the zoom. Okay, the other way. Okay. You see, it's already becoming a little bit dark and the camera has already some problems adjusting. So let me turn up the light. You see, there's also some stray light now because of the thing here. It's a little too dark. But this metallic green thing is because of the surface texture, and is caused by apparently by light interference, and it's not because it's colored green. Yeah. Do you think that the scratches on the acrylic surface will be suppressed with a thin oil film? This is actually something that is possible. I don't have oil with me now, but indeed that's I think possible. I just want to show you something else here. Also a little bit what I have here is we might also look at this. This is really old, this one over here. So let me show it to you also under the microscope. I think this is from the late 1970s. And this is also in resin, some nice butterflies. But you can see already that here on the edges, it started to turn white. I don't know if you're able to see this. Yeah, there was some sticky tape on here, and it somehow reacted with the resin. So this is a little bit an issue of these resin embedded specimens. That while the specimen is well preserved, interestingly, the resin itself sometimes suffers a little bit. The interesting thing here as well is that if you look at the butterflies, they also have become semi-transparent. So let's try to have a look at those butterflies here. I have to tell you, I did not look at this yet. Okay, so let's we might as well try this here as well. I have to change again to the stereoscopic view. I have to readjust. You know what? I need to zoom out. Yeah, but you see, and the contrast is also not the best, which kind of shows a little bit that there is the resin does have some kind of an impact here. I don't know if the refractive index of the resin and the butterfly wing are in such a way that we're actually also able to see the butterfly scales, and the contrast is also quite low. And let me refocus. Yeah, so not very good. Let me try it with the light from the bottom. The camera will try to readjust. Yep, that's with light from the bottom, and now we're able to see a little bit let me refocus here. The scales of the wing of the butterfly. Again, the camera setup does take away a little bit of image quality. Generally, you see it better, of course, while looking through it. And I see that, So stereomicroscopes are quite interesting here. Do you think that the scratches, yeah, I talked about this, like the Rosenkäfer, I recently made a decent macro image of its skin. It looks like a reptile skin. Yep, oh, that's interesting. Did anyone ever use the microscope that I posted? I don't remember exactly which ones you posted now. Try to use the Barlow lens and go to the max magnification and see your finger to see if you can see the skin cells. You cannot see the individual skin cells. The reason is because they are so close together that it is not possible to kind of separate, to see them as separate. Okay, but I can have a look anyway at my skin in any case. It should be possible. I mean, something I was not able to manage yet, but what you can try is you can put some oil over here on your little finger, and this makes your skin a little bit transparent, and then if you're lucky, you should be able to see the circulation of the blood here, because it's very thin. I've not managed this yet, but some doctors actually do that in order to test blood circulation. So I have to kind of go all the way down. Okay, and that's now my little finger here, and can see all of the different. So let's focus again, and let's go in all the way. And of course, the further you go, you magnify, the less the clarity, and you see that even when I'm shaking a little bit here. And yeah, you can also try a little bit to increase the contrast by changing the angle of the light. Yeah, when it casts some shadows, then you're able to see this better. Yeah? So you see over here the skin cells, you might be just barely be able to see them, but because they're sticking together, it's difficult to see them as separate, and therefore you're not able to see them individually. Okay, I don't have some sticky tape with me here right now, but what can be done is, as well as you can, I don't know, let me have a look here. I don't have sticky tape. You can actually, if you take some sticky tape and put it over your head, for example, and peel it off, then there's a lot of skin cells that also come off. So let me put again something interesting. I don't know, we can, if you want, we can again try to put those guys here under the microscope. Yeah, there's still. Look. Now they're not moving too quickly. Pretty scary looking pincers here. Huh? Hi from Switzerland. Since it's a stereoscoping microscope, can you create 3D images that can be seen in 3D by having them side by side, or could it be looked at in virtual reality glasses? Yes, I already mentioned this. You can take two different images using mobile phones through the two different eyepieces, or you can use image stacking software and let the software calculate it for you. So basically you take images from different levels of focus, and then the image stacking software is able to combine it together, and then you're also able to let the software calculate a red-green image that you can use to look at with stereo glasses. Piccole is the program that you might want to try out. I posted the link into the chat. Okay. Yeah, it's a bit of a hobby of mine, but mainly because I use them for macro photography. Okay. Yeah, okay, precisely, but now I've started with a whole series macro photography. Yeah, so macro photography is also, of course, quite nice. Are photo stacking and image stitching the same? No, but both of them are actually very useful. Stacking is when you're combining basically images of different focus into one image that is completely in focus, right? Because when you look at especially using high magnification at an object, so let's put in the fly again from before. So I have to find it the center here, okay? And when I have the This is especially the case at higher magnifications. So right now, the top part of the fly is in focus. I'm just going to show this to you here again. Okay, so let's look at the ear. Let's see it's a little bit too bright. Okay, but now the top part of the fly is in focus, but the wings here on the background, which are further down, are blurry. And if you want to have everything in focus, what you do is you take a series of pictures, and picture, and then you kind of focus all the way down until the wings also start to go into focus. Okay, now you can actually see a little bit the veins and the vessels of the wings. Yeah, it's overexposed now, but and then you have, I don't know, 30 different images, and you pull those 30 images into the stacking program, and then it will create one final image that is focused from the top to the bottom. And stitching is like in a panorama where you take overlapping pictures and you're increasing the size. Yeah. So this is both of them are actually used quite often in microscopy as well. Okay. So yeah, here there is the explanation already. Would I be able to study the microbes that grow on corn? Well, I'll tell you. Using a microscope, you're only able to see the shape of the microorganisms, number one. And number two, there might not be a lot of microorganisms there. So if you really want to study microorganisms and do a proper analysis, then you actually have to grow them. Because otherwise they're too few. They're too difficult to isolate. And simply by looking at the microorganisms, let's say bacteria, for example, I'm thinking primarily about bacteria right now. Just because certain bacteria look the same does not mean that they are the same. So this is a really important thing, that the microscopes are, even though they are very important, they have their limits, because especially with certain microorganisms, because certain important characteristics simply cannot be seen with a microscope. Okay, have you ever discovered a new species of microorganism to see very, very rare species? As a matter of fact, this is something which is almost completely normal. I mean, when I did my thesis in microbiology, I mean, we isolated bacteria from the air, and we characterized them. There's always something new there, right? So you do a DNA test and you discover that something is related. And if you, I mean, in my case, this was not the point, was not to discover new bacteria, but to make an environmental assessment. But this is indeed something which is by scientists done every time. And it's not even so difficult to discover new bacterial species, because there's so many of them. Yeah. Okay, the antlion has wings. Yeah. Okay. The larvae of the ladybirds, Marienkäfer, also look very strange and not at all like. Yes, that's correct. Yeah. What if the antlions move when you put them on this? What if the antlions move when you put them on the stereo? They move. So that's why they're a little bit difficult to see. Okay, because they move away all the time, and therefore, let me go again into, and you know what I'm going to do then. Oh, it's 1 hour 7 minutes already, but I might after a couple of minutes I'm going to add a little bit of water. Maybe I'm able to wash away some of the sand that's sticking on the surface. Okay, they're here. Okay, where is it? See, they're always a little bit difficult to gone. Yeah. They're running around in a circle on the side here. one antlion pushing the other one. Yeah. Yeah. Insects look like aliens under the microscope. Yep. The antlion probably was an inspiration for an alien mother. Honestly, if you look at insects and other things under the microscope, I'm absolutely sure that many of them are inspirations for some science fiction movies. Yeah. Okay. I found paramecium in some moss yesterday. Yes, that's quite well possible. Okay. I'm thinking about getting a stereomicroscope. Well, actually, I do hope to get people also interested in stereomicroscopy. Yeah. Yeah. So as I mentioned already before, stereomicroscopes are especially also suitable if you want to do very quick and uncomplicated nature observation with children as well, but not only. And honestly, there's one thing that I have to tell you. If you look at the specimens using both eyepieces, it will look so much better than here on a video. It will look so much, because you do not get the stereoscopic view in a video that you normally get when you look under the microscope. The reason why it always changes the color now is because the camera adjusts its exposure time. Yeah, and that's why it kind of, yeah. So that's the thing, yeah. Yeah. Do you have a bee? I have observed bees, but I don't have one now. And honestly, I do not like to kill bees for microscopy, because bees are a little bit rare. Yeah. So what I've done is sometimes I've found some dead wasps, for example, and I've put them, of course, also under the microscope. Yeah. The Tonlov Pro 401, I think, is a great alternative to the stereoscope if you want to film and take pictures. Yeah, I don't know it. I mean, there's so many models out there. Can you polish the resin with a Bunsen burner? I wouldn't do that, because I don't think, the reason why you probably are referring to a Bunsen burner is because you think that the plastic might melt and therefore make it more smooth. I think that this might not happen. I think that this is some kind of an epoxy resin, and I don't think that they melt. Okay, so this is on YouTube. If you're interested in making things like this yourself, and not with insects, but putting other objects in resin, there are a lot of YouTube videos out there that describe how to do that, because some people like to put things into resin simply because it looks nice for as a hobby. And there you have to mix different components, and it's like a liquid, a thick syrupy liquid, and which hardens out overnight. And so this is not made by heating it up, okay, like plastic, which melts, but it hardens out by chemical reaction. Therefore, I don't think that a Bunsen burner might work, but I did not try it out, so I don't want to risk it. Will you do a live tomorrow? Probably not, okay, but I have to say something that's really important. Next week, okay, on Saturday at the time, I have again a little time conflict. So what I would like to do next week is I would like to do a live stream at my time on Sunday morning. I'm doing this now on Saturday evening, but I would like to do this around 12 hours later. This might be better for some people, and it might be worse for some people. But the reason why I have to do this is because, again, I have some time conflict next week. Okay, I just want to clarify this. How are the E. coli in the Petri dishes? Well, they're doing fine. I have them in the refrigerator, and they were growing. Yeah, so essentially just like I showed you before. Where is the best place to find tardigrades? Short answer, please watch my previous video that I made in this channel. You can find tardigrades in moss, but not in any type of moss. I found out that the moss, which is in a forest where it's constantly a little bit moist and never completely dry, and very green, the moss, then I found many more tardigrades there than in moss that grows, for example, exposed on rocks for whatever reason. Okay, please, about the tardigrades, do watch my previous video in this channel, because I actually did make a video about that. Yeah, so yeah. Another time conflict, unfortunately. Car polish and buying wheels should clean them up. Okay. Buffering. Okay. I hope I can join next week. Yep. Have you ever felt sad for a microorganism dying? This is an interesting and an important question. It's about the ethics of the whole thing. And it's something that essentially I, as a teacher, I'm confronted with, of course, for example, when I collect insects with my students, and you have to kill the insects. Otherwise, you're not able to do a microscopy with them. And some students don't like that. We have an entomologist as well who basically supports us here. Basically, the protected insects are not being killed. Okay, so do I ever feel sorry about this? And I will tell you the following. This is a mixed thing. You have to, as a scientist, as a human being, you have to respect life. Okay? I think that's a very important basic thing. And for this reason, those antlions here, I'm not going to kill them. I'm going to put them back into the glass jar, and I'm going to release them where I caught them. Okay, not because they're protected or anything, but because I, as a human being, want to adhere to certain ethical standards. However, and this is now important, when I talk about real microorganisms like single-celled microbes, they do not have a nervous system. Okay, they don't have a self-awareness. I think other animals also don't have a self-awareness. But those single-celled microorganisms, yeah, just because they are alive does not mean that they have the same. Look, how shall I put it this way? There are so many problems in the world that we have these days. Climate change, pollution, I don't know. Yeah. Here in Central Europe, especially in Austria, where I live, is one of the countries where we have the largest land loss in the area in Europe because of so much construction work going on. Yeah, this is a really huge impact on the natural environment, pollution, all of this stuff, plastic in the ocean. And I think if we want to be ethical about something, and if we want to improve the environment, then let's worry about those problems more, because they will save way more life, animal life, microorganism, whatever, plant life, way more life than the couple of microbes that I might kill on my microscope slide. Okay, so as a matter of fact, and I also tell this to my students, I consider it even ethically problematic to worry about single-celled microbes and bacteria on my microscope slide if I don't also worry about other things. So I cannot, yeah, so this, I think this is a really, for me personally, you see I'm getting emotional, an important thing is let's be ethical about the right things. And I'm not concerned about microorganisms dying on a microscope slide, because I'm more worried about the massive loss of biodiversity that's happening right now. Yeah, because that is a real problem. By the way, microorganisms dying, if I eat yogurt, my acid in my stomach kills off billions of bacteria. Yeah, so I don't feel bad about eating yogurt either, right? So okay, but let's go back here to microscopy. If you want to polish them, you can polish it with toothpaste. I'll try that. Okay. Yeah. The rarest event you've seen under a microscope, there are not so many rare events, but occasionally you do see some microorganisms eating other microorganisms. Yeah, I think that's kind of also a little bit interesting to observe. Okay, so yeah, that's a little bit the thing. How are tardigrades able to withstand such harsh conditions like space and extreme heat and cold? Is it possible to kill them? Of course it's possible to kill them, and they can be killed. And as a matter of fact, tardigrades are not very resistant to heat. According to some research I read, they're resistant to drying, but once it becomes too hot, they don't like it so much. Apparently they have certain chemicals in their body that are able to preserve them if they lose water. Yeah, so this is a way of kind of surviving also, for example, the changing water conditions, yeah? In moss, for example. Yeah. So what I'm going to do now is I'm going to try to remove a little bit the water, not the water, but the sand from those how do you call them, ant lions, by adding a couple of drops of water here and see what happens. I don't know if they like water or not. I mean, they're exposed to rain. Okay. Yeah, and you see, that's interesting. Yeah, some of the thing is washed off. Some of the sand is washed off. Okay. Yeah, because if there is generally it doesn't like it if we shake it, but you see, it's moving away again. Yep, but it's kind of evident that the water itself, it does not wet. Wetting means is that the surface does not seem to accept the water very well. I'm always moving off this out of the circle here. I have to make. So if you put water on top of it, apparently, you see, it runs off a little bit. Yeah, see, it starts to swim. Yeah, yeah. It seems to be a way of keeping itself dry. Yeah. That's also something interesting that I learned. Yeah. Yeah. Yeah, and that's actually maybe one of the reasons why so many, some insects are kind of a little bit hairy, because this is allows the water to kind of run off. Huh? So yeah, so we can see that yeah, I'm not able to remove the water, the sand from the surface quite easily here. Yeah, huh. Yeah, it's going around in a circle. Yeah. Yeah, so, but I think what I'll do now is 1 hour 19 minutes. I'm also starting to feel hot and tired. Here's a second. Here's the second one. Yeah. So let me quickly go through the remaining questions here. How are the tardigrades able to withstand? Yeah, I'm right now looking at tardigrades from moths with a stereomicroscope to collect a few of them. Most of them are dead for some reason. It could be that tardigrades also do molt, I think. Of course they molt, and this basically means maybe you're not looking at dead tardigrades, but maybe only at the skin of some of the tardigrades. This could also be a reason, okay, that essentially they're empty, just the empty skin of the tardigrades. You've been alive for a long time, yes. Yeah, okay. It is interesting to see the antlion's head has the same basic shape as the adult insect, whereas the rest of the body changes a lot. Yep. Yeah, interesting that you noticed that. I didn't see this. Yeah. You see, it's kind of difficult here. It's kind of always moving away. Yeah. Yeah. So, but what I'm going to do is the following. Folks, I think I'm going to leave it at that. I'm going to return the antlions again into the jar, which is over here. Where's the jar? Here I've got a little jar with a little bit of sand, so I'm going to return the antlions into the jar here. I'm just going to touch the antlions. Yeah, I've touched them, actually had them already on my hand. They don't bite. Yeah, so what I'm going to do is I'm just going to leave it at that again. I'm going to, yeah, simply, I would like to simply motivate you again to also make some videos or some articles or whatever, and add the following hashtag. And maybe in August, when yeah, we've got 300-year anniversary, we're able to watch a few videos of what people created simply by Googling for this hashtag. Yeah, so it's kind of like a decentralized little project. Yeah. A short reminder, next week, not on Saturday, but on Sunday. I might go back then again to the Saturday, but occasionally I do have some time conflicts. Okay, and I wish you all the best. Hope that you liked the live stream. I just realized I actually did not put the spider under the microscope yet, this gigantic spider, but honestly, it is so big that you're not able to see a lot anyway. Okay, so yeah, so sometimes not all specimens are quite suitable. So in this spider here is simply too big. But I'm going to leave it at that. Yeah, all the best. Happy microbe hunting as always. Yeah, and see you around next time. And please, if you have not done so, do subscribe to the channel here. Yes, almost forgot. A big thank you to all of the supporters, to the Patreon supporters. Big thank you to all of yeah, you who've been participating in the chat and have been asking questions. Yeah, and yeah, I'll of course normally also keep on uploading regular videos as well. See you around next time. Bye bye.